Hello, my name is Francisco, and I'm a postdoctoral researcher in the OncoRNA lab from Ghent University. Together with my supervisor, Professor Peter Mesdag, and other collaborators worldwide, we want to understand why certain patients respond to immunotherapy while others do not. In a nutshell, immunotherapy consists in helping our immune system to recognize and eliminate cancer cells. While it is very promising, it's very costly and has been proven to be effective only in a minority of the patients, unfortunately. The underlying reasons why some patients respond to the treatment while others do not remain largely unknown, so that's where my project comes in. We will not focus on the cancer cells, but on the immune cells that are present in the patient. As you may know, a tumor is not only made of cancer cells, but also contains other cell types, like cells from our immune system. Research has shown that the number and composition of these immune cells differ strongly between healthy people and cancer patients, and even across cancer patients. We believe that these differences in immune cell composition are an important factor in defining the susceptibility to immunotherapy, and therefore I want to investigate them in this project. Imagine you are in a blind tasting and are giving, on one side, a praline to try, and on the other side, each individual ingredient present in the praline. You are then asked to provide the percentage of each ingredient in the praline you tried. While for us, human beings, this kind of question is not that easy, powerful computers can actually do this in a very exact way, using a technique called computational deconvolution. In such a setup, the computer is first trained to recognize each individual ingredient. So a bit like having your computer first try chocolate, then butter, and then sugar. Afterwards, you let it taste a whole praline, and it can calculate the exact proportions of each ingredient. Of course, in our project, it's not about a praline and its ingredients, but about a blood sample and the different types of immune cells it's composed of. In an analogous way, as with the praline, we first have to teach the computer what types of immune cells exist and how they look like, so afterwards it is able to retrieve the exact proportions of the different immune cells when we feed it with a complex blood sample. By using this technique to analyze the blood samples of several patients, both responders and non-responders to immunotherapy, we hope to identify the differences between them and assess whether differences in a specific cell type can tell us whether the patient will respond to immunotherapy or not. In conclusion, with the help of CRIG funding, we hope to find a way to predict whether or not a patient will respond to a certain type of immunotherapy, based on the assessment of the immune cell composition in a blood sample. This will help oncologists to select the most effective treatment, gaining time to exploit other therapeutic opportunities for the non-responders. Moreover, this will limit their exposure to toxicity, and it will also reduce healthcare costs.